Hello, and a very warm welcome to today's Select Science webinar, Emerging Techniques for Improved Biomedical and Clinical Research. My name is Charlie Carter. I'll be moderating today's presentation, and I'm delighted to be joined by Dr. Francesca Palombo and Professor Nick Stone from the University of Exeter. Please feel free to submit your questions for the Q&A session at any time during the webinar. Without further delay, I'd like to hand over to Francesca and Nick.
Hello, and thank you for the opportunity to share my thoughts about an emerging tool in biophysics and mechanobiology. I'm Francesca Palombo from the University of Exeter, and I'm going to talk about emerging Brillouin Microscopy in biomedical life sciences. So in recent times, there have been a number of high-impact publications in this field, and for those of you of interest, please look at this impactful review papers on this new technique, and also there are some perspective work and opinion pieces to look at in terms of the field of optical elastography, which is emerging as we speak. So in Breon light scattering, what is this technique? Similar to a palpation test, this technique involves the use of light to investigate the interaction with refractive index fluctuations, which are caused by the propagation of thermally driven acoustic waves of phonons in the material that probe the acoustic landscape in terms of compressibility and viscosity on a micro scale. So you see that the interaction causes the shift in the frequency of light emerging from this interaction, and spectrum that we measure is in the gigahertz frequency range. So we are very, very close to the elastic light in green, and also it's a very, very weak effect that poses some constraints in the quality of the setup, as I mentioned later. So from this measurement, you retrieve the V. V is the speed of sound, and from the speed of sound, the complex longitudinal elastic modulus with its components that are the storage and loss modulus reported in these expressions. So first pioneering works in the late 1970s have already demonstrated that acoustic phonons probe the elastic modulus, so elasticity of biological materials, but soft matter in general. So the important information here is that temporal scales in Brillouin spectroscopy need to be considered. Depending on the actual state of your material, say if you are in a liquid or gel, in a solid phase, we are looking at different curves of this dispersion in terms of M prime and so the storage and the loss modulus. And when we look at the frequency scale here, we are obviously sensitive to the shift in this structural relaxation that is inherent to the material itself and depends on the hydration state, on temperature, and so on and so forth. So temporal scales, but also spatial scales are important when considering acoustic phonons. That is the probe. It's not light, the probe. The probe is the acoustic wave. So there are at least three relevant scales that we are concerned with. The smallest scale is the Phonon Wavelength that is dependent on the wavelength that you use for the illumination. So we are in the range of 200 or 300 nanometers in our applications. But most important is actually the propagation length of the phonon itself, this L, that irrespective of the actual size of your scattering volume that is governed by your optics, this L actually regulates the extent of the signal that you measure. So you can end up in a very counterintuitive but interesting situation whereby increasing the optical resolution that in fact is reflected in a point spread function that for Rahman indeed increase in terms of resolution, but the elastic resolution actually worsens. And this, in fact, depends on the material itself. The other consideration to be done is that there are different types of moduli. We are all familiar with Young's, shear, and bulk moduli that are measured through quasi-static experiments, and they are expressed, or they are related to each other through this expression. In Breon, we measure the longitudinal elastic modulus through high-frequency measurements in the gigahertz range, as I've shown. And the propagation of these compressive waves through the material, if you think of illuminating from above, yields the M that is related to bulk and shear modulus, but to a lower extent, if you like, to the point that we expect M to have smaller gradients across the material than the Young's modulus has. So values are in the gigapascal range, and the gradients are smaller. Brillouin spectroscopy poses some restrictions, as I said, to the quality of the filter in order to analyze the light that emerges from this scattering event. The traditional approach is to high contrast tandem Fabry-Perot interferometers. And in recent times, we're also looking at non-scanning single echelon VIPA spectrometers that provide a faster approach to the measurement of these spectra from an hydrogel system. And you can see in linear and in logarithmic scale how these peaks look like in Brillouin. We first introduced the combination between Brillouin and Raman microscopy from the same scattering volume in order to associate molecular specificity to the resonance that we measure in Brillouin that is non-specific in terms of chemistry. So this is very interesting because you can understand the origin of these acoustic resonances in terms of the molecules that make up the system. We also first introduced the new generation of high contrast tandem Fabry-Perot interferometers a few years ago with unparalleled contrast for the analysis of turbid media such as biological samples. In this type of implementation, the avalanche photodiode is the detector that provides increased performance over more traditional implementations. So some recent results from our lab involve the study of tissue phantoms in order to understand the information content of the Brillouin spectrum and also the significance of these signals in a biomedical context. And you see that by increasing the concentration of the polymer in these gelatin hydrogels, you indeed are able to increase the rigidity through the measurement of the Young's modulus. And in parallel to that, we observe an increase in stiffness measured by the storage modulus. But most importantly, we observe an increase in the loss modulus. So the most significant changes here are in the line width of the peak. So it's very important to be able to retrieve the, let's say, true the spectral shape from the spectrum, and to measure this type of effects that are reflected in the viscosity rather than the stiffness, more in the viscosity. And this is due to the interaction between water and polymer that enables the relaxation of water dynamics of the interface to be retarded across concentration range. As I mentioned before, there are developments on the spectrometer side that enable non-scanning implementation to achieve faster acquisition rates. So this is through, for example, VIPA echelons, firstly introduced in the 1990s and then applied in Brillouin imaging for the first time about 20 years ago now. And in a two or multi-stage VIPA configuration by Giuliano and Andy in these interesting papers. So with this setup that we have implemented in our lab, we're looking at using the green laser, a green diode laser, combined with an inverted microscope, and the scattered light here is analyzed through a two-stage VIPA, as you can see, and there are optical masks to select the peaks that are analyzed and measured through an SIMOS camera. So the camera is an imaging camera because we obtain a readout that is based on extracting a spectrum along the diagonal. Here is the dispersion axis. So the measurement lasts up to a few seconds, but we can go much faster depending on the obviously the camera, but also the sample itself. So here are some recent data from testing that was enabled by Andor on the SIMOS cameras and EMCCD cameras back to back. In terms of signal to noise ratio for water, what we observe is that perhaps we can look straight at the final plot in here. You have the best performance, although with large error bars here, but best performance comes from the sonar, so the latest SIMOS camera, compared even with EMCCD. The same, in terms of standard error on the frequency shift that we measure. So this is looking at the repeatability, if you like, of the measurements. Again, you have best performance from the sonar camera compared to the others, and more interesting for us is also the line width retrieval, and again, sonar seems to get better. Of course, you get if you can increase your dwell time, obviously you get closer to what this line width is. But obviously, sonar is definitely the best performing one. But bear in mind that with EMCCD at high gain, you can achieve very fast acquisition rates of the order of milliseconds, while for the others you need to go more like hundreds of milliseconds, as is shown in this slide. So as I said, careful to the shape of the peaks, because we also observe a different intensity inversion here in the case of EMCCD compared to the SIMOS cameras on the top. So top of the list, I would say at the moment would be the new generation SIMOS cameras. So alongside this technical development, there have been also a range of applications that have been demonstrated for this Brillouin imaging technique. Top left, atherosclerosis in animal models. Bottom left, cell spheroids or cancer applications, and also in tissues, this is melanoma, but also in dementia, as we've shown in our papers, for the amyloid plaques, also applying machine learning to segment the Brillouin peaks in the case of hyperspectral images. In life sciences, the notable applications have been in single cell imaging, here in the mouse fibroblast cell or plant cells, and also in entire embryo. This is a murine embryo, but also zebrafish and live nematode using stimulated Brillouin microscopy that has been demonstrated this year. In terms of clinical translations, important has been the application, the first in vivo human application of Brillouin using a 780 nanometer laser through the scanning of the cornea, which has been then that has led then to a clinical trial, large cohort clinical trial for detection of corneal degeneration using Brillouin at the Massachusetts General Hospital. So I'm going to finish off by saying that this is an emerging technique that probes viscoelasticity on a microscale, provides a new mechanism for achieving contrast and a new spectral window in biophysics, mechanobiology. There are important considerations to be made in terms of spatial and temporal scales, depending on the material of interest. But also we believe that molecular specific information is important for this technique, and therefore we encourage the use of Rahman, for example, as a correlative technique. Something to be careful: laser wavelength, laser quality, also the scattering geometry that you use. There's not only back scattering geometry in 180 degrees that I've shown here. Very important is the quality of the spectrometer, depending on the sample. And of course, a fast and an accurate camera, and most importantly, most sensitive camera for this kind of applications. So let me thank all of my collaborators, the people who've performed the experiments, so Michelle in particular, but also Nick Stone and Peter Wheeler of Exeter, and people in our collaborators' lab at Perugia, all the funding bodies, and thank you for listening.
Thank you, Francesca. So I'm going to follow on from Francesca. My name is Nick Stone. I collaborate and work closely with Francesca at the University of Exeter. I have come out of the NHS as a medical physicist, working for many years exploring the use of light for both diagnosis and therapies for early cancers. I'm going to talk about Raman spectroscopy, and it's a complement to Brillouin. It's looking at inelastic scattering of light, but from the molecular vibrations that are induced in the materials that we probe. So, following on from the concept of Raman, I will explain the concept of Raman as we go. And so I want to put it into context really. So as somebody who's worked with clinicians for many years, recognise the importance of early diagnosis, and it's vital that we catch a disease before it becomes symptomatic, and this is usually the case when there are a lot of cells and the tumour is starting to invade into the body, which is very hard to treat. So if you can find it early, you can treat it more effectively. The problem is, to find it early, you need to have a group of people that are probably asymptomatic but at a higher risk of getting that disease, so you can potentially surveil them. Now this is Hugh Barr, a leading surgeon at Gloucestershire Hospitals, who has spent a lot of his career developing methods for identifying early cancers in the esophagus, which can be a really significant disease and leads to many people dying early from that condition. To be able to make a diagnosis, this requires going down with an endoscope, looking for invisible disease and tearing out lots of bits of tissue, which end up having an esophagus, which can have lots of holes in it, but then leads to a pathologist having to look at all of those different samples. And quite often, there's an image missing here where the text is on the bottom right, but quite often the pathologist will look at many tens of samples from each individual, and most of those will probably be normal. So this is still trying to find disease in an invisible disease, which is very, very difficult. So in addition to that, the pathologist, as well as having to deal with all the samples, we're missing large areas of the esophagus, but also pathology has a great deal of difficulty making a decision for early disease. I won't go into all the detail here, but this is basically showing that pathologists don't actually agree on each of the samples that we're looking at in those 6 images of histopathology, the H&E stained images. They don't actually agree on any of them apart from B3, which is clearly an issue. But they do agree regularly on obviously very normal esophagus and highly cancerous esophagus. It's the bit in the middle where we want to be able to make the early diagnosis and make the accurate treatment, the high quality treatments. So if we consider the use of Raman spectroscopy, where we shine laser light or monochromatic light on the sample, we are looking at the inelastic scattering of light rather than the elastic scattering of light, which is more common. It's a much weaker effect. It has a low cross section, so we need very sensitive detection methods and very sensitive optics to cut as much light as possible. But in effect, we're looking at the scattering of light off the molecules of interest, and we end up with a fingerprint of the biomolecules found within the cells and tissues. That allows us to effectively develop a fingerprint by looking at the laser line would be at 0, and we're looking at the shift as we move across to the right along the bottom axis we get a fingerprint of the different materials that we're measuring. And this is an example of 4 prostate cancer cell lines, and we're able to separate those out by their individual biochemical components. Here's an example of tissues collected from lymph nodes, and I will talk about those later, but you can see their fingerprints and they separate as well differently. And you can also build up images and maps to create pathology based on spectral information to potentially support a pathologist making decisions. So back in the early days in this field, of 20 years ago or more, we were looking at the fingerprints of disease under a microscope to try and understand what they would tell us, and to understand whether we could extract key features to separate out the disease. We recognised that we needed to use near-infrared light, and I'll come back to the wavelength choice later. And that, in this example, we found that 830 was best for esophageal tissue to reduce the fluorescence that we see at lower wavelengths. By extracting by using multivariate statistics, we can, or machine learning approaches, we can use the fingerprint of the spectrum that we measure from each individual point. So each dot in this scatter plot on the top right is a spectrum that we've measured from the esophagus, and we're creating a principal component fed linear discriminant analysis plot, which shows that we're able to separate out the different pathology conditions. And we can do that pretty accurately. But if we do this by training our models with a consensus pathology opinion using 3 experts, and this has enabled us to have an accurate result. Now, if we move to the bar charts we see, the one on the left is basically demonstrating how well we can separate all the pathology groups in the esophagus. And you'll see that this is pretty effective, although there is some small crossover in some of those bars, and that is using the consensus panel a pathology opinion. Now, the next bar chart to the right of that is using the majority opinion, where 2 of the 3 pathologists agree, but one of them disagrees. And you can see straight away this completely causes a detrimental, has a detrimental result on the pathology prediction. So it's really important to use a really strong gold standard if you're going to use novel methodologies for cancer diagnostics, particularly in the early phase of disease development. Okay, so over the last 20 years, our group and many others have explored vast parts of the body, vast parts, I should say, a myriad of areas of the body, and it's been demonstrated that we can pretty much discriminate different phenotypical expression of disease throughout the body in most areas. For example, in the bottom right-hand corner here, we're showing the fingerprints of the pathology groups in the bladder and how well we can separate that with pathology. So Raman Spectroscopy has been demonstrated to effectively be a diagnostic platform technology, at least in the laboratory space. It is rapid and non-destructive. We're just using low-power laser light to measure this molecular vibrations that will tell us what are specific to the molecules present. We've demonstrated over a number of different studies where we can provide reproducible signals between systems and centers. And we have demonstrated that if you train it with a consensus of leading pathology opinion, you can exceed the performance of independent pathologists. In addition to that, prognosis appears to be possible by looking at follow-up of patients and seeing whether their conditions check, whether signatures are different relating to those that develop cancer in the future or not. And that has some examples we've shown in the past. Okay, so what we're doing at the moment is working towards an in vivo confocal measurement of cancerous of early cancers in the esophagus. We're using a miniature confocal endoscopic probe that we've developed as a team between Gloucestershire Hospitals and the University of Bristol, with John Day leading on some of the design and build of that, and Hugh Barr leading on the surgical side of things. And what we're trying to do is take a measurement of the lining of the esophagus to provide a measure at the surface of the esophagus, not at depth. The confocal part allows us to look at the very surface and then provide the surgeon with an indicator of the likelihood of disease. That's an ongoing study that we will report on next year. Now, we looked to go down the endoscope initially because we were worried about the condition of Barrett's esophagus and leading on to high risk of esophageal cancer. But by modifying or playing adjusting the different parameters you can use for Raman systems, whether it's a microscope, whether it's looking at histopathology approaches, whether you sit next to the patient with a system that enables you to do intraoperative analysis of surgical resected samples. This is lymph nodes from the breast we have here, or whether we go closer and closer to in vivo, minimally invasive approaches using the endoscope, like I mentioned, or potentially using a smart needle or transillumination, and I'll get to that later. So we're currently developing a smart needle for use in head and neck cancers, and where we have lumps and bumps that present. These are usually swollen lymph nodes, and those swollen lymph nodes need to be removed quite often to see whether, for certain, whether there is a cancer present or whether they're reactive lymph nodes, meaning they've swollen up and haven't gone back down after an infection. And so if we measure the fingerprint of those, a lot of this work was done by Linda Orr, who was a head and neck surgeon doing a research degree with me in the past. She was measuring all of these different spectral fingerprints, and if we use those to construct a diagnostic model where we look at the scatter plot in the bottom right, each of the dots in that scatter plot represent a spectrum from lymph nodes from the head and neck, and the green ones are reactive nodes, the blue ones are lymphomas, so they're primary cancers, the black ones are adenocarcinomas, and the red ones are squamous cell carcinomas. So the red and the black are secondary cancers that are basically appearing in the lymph nodes, where there's a primary cancer somewhere else nearby. So you can see that Raman spectroscopy has a great deal of power for discriminating disease, but this was all done under a microscope. And so what we're trying to do now is remove the need for taking out those samples and provide an intelligent optical biopsy on the end of that smart needle to enable a real-time measurement of that disease condition. And some of our very early work, John Day from Bristol, as I mentioned before, and I did some work where we made a very small needle probe system which enabled us to take measurements from lymph nodes on the end of a needle. These are receptive, but they're very distinct signatures that we see between metastatic and benign nodes. So we're currently developing a needle which is slightly more complex than that, one fiber in and one fiber out, and we've now got a fiber with a probe with disposable tips and multiple fibers, and we're working towards the first in human studies in the lymph node at Gloucester hospitals. So to achieve this, we have a system where we have a single illumination fiber surrounded by 6 collection fibers. And we've done a study already where we've demonstrated that the signatures that we measure in a short space of time enable us to give a reasonable discrimination. It's not perfect, but as you see, we have quite a large range of pathologies that we're measuring from resected lymph nodes here. We're plotting out the receiver operator curve, and that if it all went up to the top, this is in the top right corner, the top right plot, I should say. If it had an area under the curve of 1, it would be a perfect test. If the curve went along the dotted line, it'd be the same as chance. So you can see it's a pretty good test, but it could always be improved. And we're working to optimize that as well as we can before we go in vivo. So thinking of how we actually use the system. So in some of our early prototypes, you can see there's lots of fibers here and all the like, and it's quite important that to be able to get a sensitive measurement, that we're able to measure spectra using a camera that is able to both measure the spectra from an array of fibers, but also so it has to be sensitive to near-infrared light, and it also has to be able to look at an array of fibers and ideally separate the signatures from those, which we have been able to do using this IDIS deep depletion camera. We're looking at how we might be able to modify that for its use beyond the head and neck into potentially all sorts of areas of the body. And in this example, we've had a test phantom where we're basically measuring spectra as we insert the needle into the sample to the bottom right. I'm talking about here, and if you see that the tiny little peak within the red, well, it's not a circle, but within the red ellipse, you'll see that little peak is representing hydroxyapatite. So we've been able to see some calcification material that is buried within that phantom breast that we're using. And this could be very useful. Now, we've been working with Tom Hubbard, who's a breast surgeon at the Royal Devon and Exeter, and he also did an MD in my team, and he's been exploring the application of Raman spectroscopy to measure the water content within breast tissue. And other groups around the world have demonstrated that water can have an important change. Water indicates the presence of breast cancer. If you look in the MRI scans you're seeing on the left-hand side, you can see the water content is significantly greater in areas of breast cancer than normal breast tissue. Owen Pupples and his team in Rotterdam have also, sorry, I clicked on accidentally then. Owen Pupples and his team in Rotterdam, let me go back to where we were, something went wrong there. So in the top right corner, Owen Pupples and his team have demonstrated that oral squamous cancers also represent a very significant change in water. And this is the high wave number part of the Raman spectrum. And what we're seeing here is the relative water content to the protein and fat pH peaks, which you see at around about 2985 wave number. So you can see that the water is significantly higher in tumorous oral cancers, but you can also see the same kind of effect in breast tissue if you look at the spectrum below. So what Tom was trying to do was see how he could measure these signals, and we were looking at using different wavelengths of light. And if we use the 680 nanometer illumination, it was quite possible with using a whole range of different phantoms. You can see these are intralipid phantoms on the bottom left, and these are gelatin phantoms. Sorry, they've all got gelatin in, but they're intralipid as in adding lipids, and just water and gelatin on the right. And what we're trying to do is demonstrate that we could see the differing water content when we varied its concentration, and that worked very well with 680 nanometers. And the reason, and I'll come back to the reason why we're using 680. The problem is, if you look at the tissue, the meat samples on the bottom left, when blue dye is present, it causes a significant fluorescence, even in the high wave number region. And this is a big problem because breast surgeons inject blue dye into the tumorous region so they can find out where the tumor drains to which lymph nodes. And this can cause some problems. So what we tried to do then is use a wavelength where we would reduce that fluorescence, and we've considered using 785 here, which can do the job. But what you'll see is that the water peak relative to the CH peak has basically pretty much disappeared in the intralipid spectrum, and is pretty noisy and unpleasant in the gelatin with water spectrum. And this is all down to the quantum efficiency of the detectors we're using. So we use very effective deep depletion cameras under normal circumstances when we're looking at the fingerprint region of the spectrum. But when we move across to the high wave number region, you'll see, if you look in the QE curves on the left, you will see that if you follow the red line along, that's effectively our camera. So if you use 680, we were still able to measure that band is basically that I've put in blue, shows you where the spectral ranges that we were able to actually measure here. And the 785 one is what would happen if we were using a 785 laser. So you can see why our sensitivity and our ability to measure the spectra has dropped off significantly. So what we did to enable us to look at blue stained tissue, with 785 light, which doesn't induce the fluorescence, we've had to move to an InGaAs camera, which is also available in a similar configuration. And you can see on the plot on the right, we've got a much higher sensitivity to the 785 nanometer light. So Tom, as part of his thesis and has submitted the paper, which is just under review, used 785 nanometers. And what he was able to do was demonstrate that there's quite a significant difference in breast tissue. These are all frozen breast tissue samples taken from patients during surgery with ethical approval. And looking at 192 samples, he was able to separate out tumor from non-tumor very effectively, based purely on the water. In addition to that, he was also able to show that the presence of the blue dye, so the ones with high blue dye are the blue bars in the bottom, and the orange bars are those without blue dye. And you can see that the non-water effect between the tumour and non-tumour is still significantly different, even though the blue dye may or may not be present. So that's an important step forward to enable this to be used in breast intraoperative analysis of breast samples. Okay, let me finish off with a quick canter through the most non-invasive approach of using Raman spectroscopy. Ideally, if we can not insert anything into anybody, we can have a very amenable treatment that most of us would be very happy to have. Sorry, treatment diagnosis most of us would be happy to have applied to us. The problem is getting light through a large volume of tissue in the body is quite tricky. However, using methods such as spatially offset Raman spectroscopy, invented by Professor Pavel Matusek at the Rutherford Apton Labs, who I've worked with since around about 2005, we've developed a number of approaches where we can look at the spatial offset of the illumination from the collection point, and this enables us to look deeper and deeper within the tissue of interest. If we go to extreme cases where we look from one side, we illuminate on one side and collect from the other, such as what is being done here on the professorial thumb, I'm measuring basically the bone signal, which I'm highlighted by the hydroxyapatite there, but I'm measuring the skin, the nail, and the bone of within my thumb by shining light straight through it. Now, obviously, this could be effective if you just wanted to look at the skin or the bone, like I'm showing you there. But there are many, many applications where we'd like to look deeper than that, and where we can have quite an effect potentially on the diagnosis of disease. Now, in breast cancer, there are 2 million women with mammographic screening every year, and around about 100,000 are brought back for further tests because calcifications are found within those breasts and that require biopsy removal for pathological discrimination of disease, identification of disease. Those individuals have type 2 calcifications, which are calcium hydroxyapatite. And in this example here, we've demonstrated by using these spectra that we have along the bottom are measured through a large piece of pork joint with an insertion of calcifications within those. This is having protein-rich and fat-rich and skin tissue, very much like a breast phantom, and demonstrating that we can separate the type 1 and type 2s effectively. But in addition to that, in other work that we've done, published in Baker et al. British Journal of Cancer, we demonstrated that the signatures that you see or the composition of calcifications change as you move from benign in the green down to more advanced cancer in the dark red. And that really demonstrates that we could potentially measure that compositional change by looking at the Raman spectrum itself, if we were able to resolve it sufficiently well. So we're in the middle of a study where we're creating an in vivo system for the first in human studies using this approach. And it's vitally important for us that we collect every tiny fraction of the light that's available to us to maximize this throughput. We're trying to shine light through 5 centimeters of breast tissue. And so we need the most sensitive cameras, and we need we need to be able to spectra at as much light as possible. So what we do is we fill our spectrometer to the max, we open up the slits, we use a high dispersion grating, and we want a camera that's able to measure as much, as many of our fibers or as many of our beam as possible to maximize the signature that we're able to measure. So all of those studies are ongoing. We have 3 first-in-human studies funded and ongoing, as I mentioned. There is a high potential of Raman being applied in the clinic in the future, but there is still a long way to go. There are a lot of hurdles, but the principle there is that there is a high potential of some of these delivering to being something particularly useful, particularly if there are applications where there are no better methods. But Raman's strengths really come in the fact that we're measuring the molecular changes of disease, and this enables us to potentially predict the future structural changes that will result in symptomatic change. So we expect to be able to make a diagnosis earlier than something that ends up with a structural change. We're currently part of a study, a grand challenge funded by Cancer Research UK, which is looking at the signature, the Raman signatures of the breast calcifications. And we, at the early stage, appear to be able to indicate the likelihood of individuals going from ductal carcinoma in situ, picked up at mammographic screening, which is the non-invasive cancer or condition in the breast, identifying the difference between those patients that go forward and develop invasive cancer. So keep an eye on the work that we develop, and we'll be publishing that next year as well. So thanks everyone for listening, and thanks for all my collaborators over the years who have worked closely with us, both the clinical teams, the biologists, the chemists, the physicists, and the computer scientists, all part of this wider team, able to deliver on this kind of work. And for those of you who are interested in this kind of area, ClearSpec is the International Society for Clinical Spectroscopy. It's a great community that brings together both clinicians and scientists for further discussion in this domain. Thank you all for listening, and hope to answer your questions later.
Excellent. Thank you both for those brilliant presentations. It's now time for the final part of today's question session. It's the Q&A. And can I please just ask that you turn off your event audio, Francesca?
Yes, my audio is on, I think. Yeah. Can you hear me?
Yes, we're just getting a little bit of an echo. So if you stop your event test audio.
I did stop it.
Ah, okay.
Sorry.
Is that better? Excellent. Okay. Apologies for those listening in. I had a bit of a technical difficulty there. But those presentations were excellent. So thank you for that, both of you. It's now time for our Q&A. So I have a few questions for you, Francesca, to start. The first one is, what is the relevance of Breon signals in biomedicine, i.e. high frequency versus low frequency measurements?
Yeah, so the relevance is such that what we measure is a spectrum that when you look at the peaks, the Brillouin peaks, their frequency shift is related to the longitudinal modulus. This is a mechanical property. In fact, based on the frequency shift, say high frequency of a material means stiffer, low frequency means more squishy. So it's a measure of its mechanical properties. And it's different from what you can measure by quasi-static measurement. So you do these experiments using gigahertz acoustic waves. So if you like, it's a high frequency technique compared to others, for example, compressive testing and so on. But in fact, also the modulus is different. So you're looking at a scale of frequencies that obviously when you have viscoelastic materials such as the biological samples, the mechanical properties changes with frequency, but they also change when you look at different types of elastic moduli.
Excellent, thank you. We have another question for you, which is there a relationship between tissue function and Breon-derived elastic moduli?
Yeah, so we know that mechanical properties of cells and tissues are related to their structure and their functions. So what we measure, as I said before, is a mechanical property, which is the stiffness tensor of the material. And therefore that relates obviously to the structure and to the function of that particular sample, may be like a cell or particular tissue.
Lovely, thank you. and we have a couple of questions for you as well, Nick. So an attendee is wondering whether you can do Raman imaging in vivo.
Thank you. Okay, so Raman is a reasonably weak effect where probably 1 in a million photons are inelastically scattered, and that does make it quite difficult to create images using standard approaches. So unless you use coherent Raman imaging, which is quite difficult along the end of fibers, usually the method is about making very, very precise molecular compositional measurements at a particular point. So images are difficult. Maybe mapping is the best you could get, slow mapping.
Excellent, thank you. And another attendee says, you mentioned the use of water as a marker for cancer. How specific is this likely to be?
Okay, so water is clearly something that is apparent in tumours, in reasonably large tumours where there's leaky vasculature, which means that the blood vessels aren't formed appropriately and they tend to leak out and not drain the tissue particularly, which is often why tumours feels quite hard when clinicians try to find them. They can feel the lumps. Now it is that things like cysts, fluid-filled cysts, will also have a lot of fluid in, so they're not entirely specific to cancer, and they would probably be only specific to tumours that have got to a reasonable size. So I think it's much more about a tool when you know somebody has a cancer present and you're trying to locate it with accuracy, spatial accuracy, then it's a very useful tool. Maybe as a first diagnosis for early disease, it's probably not the first part to go to, but using the fingerprint would be the appropriate approach for that, I would imagine.
Excellent, thank you. So Francesca, one attendee is interested to hear your thoughts on whether this technique can be used to map the spatial distribution of cross-link density in transparent materials. And they also want to know what the spatial resolution is and how deep these structures can be recognized below the surface.
Yeah, I think there are a few questions there. So if I'm correct, so starting with the first, whether we can map the density, right, of cross-links. So yes, the signal that we measure depends on mechanics, as I said, to the elastic modulus, as well as on density and refractive index. So you can determine density provided you know the other two parameters. On the other hand, this relates to the other to another question that Fernando asked in terms of resolution. So if you're looking at point spread function in comparison with Raman. So Raman is so the probe is very local, is the molecular vibration. So the point spread function reflects the actual focusing optical resolution, if you like. On the other hand, in Brillouin, what is important to understand here is that the probe is not the light, but is the phonon. And the acoustic phonon has a length of propagation that depends on the sample and depends also on the focusing optics. So so it's not straightforward, and really the application of the two techniques really get you closer to what density distribution you want to measure in your material. I'm not familiar exactly with the materials that the person is pointing at, but in general that is the case. In terms of thickness, so the thickness, as I said, better to use, for example, Raman to determine the actual thickness, because as I said before, point spread function is actually matching your optical resolution. In terms of I don't remember the second question. Sorry, can you ask it again?
Yes, of course. So the follow-up question was, what is the spatial resolution and how deep within these structures can be recognized?
Yes, thank you. So the resolution, as I said, so it depends on the actual length of propagation, the mean free path of your acoustic phonons. That depends on the sample. Okay, so this is something to be measured and to be understood. And when you are hitting an interface, say because you have a certain thickness, right, and then something underneath with a different, let's say, chemical, structural property, you would imagine a shift in your Brillouin peak, and at that point you're sure you're crossing an interface, right? And so you have a measurement of the thickness that you can certainly compare and contrast with other techniques that I mentioned. Raman will give you a better close match, let's say, to the optical resolution.
Lovely, thank you, Francesca. We've got a question for you here, Nick, and that is, are the Raman fingerprints identified and classified purely by AI methods? And is there any model or other way to predict Raman fingerprints from molecular content?
Thanks, Charlie, and Fernando for the question. So, okay, so there's some complexity here. The easiest, because there is not usually a clear Raman peak for specifically for the disease. It's usually a combination of biochemical compositional changes that, rather than rely upon direct fitting of specific biochemicals, it's usually more accurate to develop an AI type approach or a multivariate statistical analysis that takes into account the majority of the information in the data. But if you use appropriate methods such as linear methods and things that you can actually look at, you can actually extract the information that the decision is made upon. It's much easier to then understand what the biochemistry is behind the decision making, and that's what we tend to do with things like linear discriminant analysis. You can obviously apply more advanced AI approaches, which can get a better result in terms of performance of discrimination. But there are alternative approaches. So there have been some by our group and by Michael Feld's group at MIT, who I'm afraid is no longer with us. But his group and ours have both took approach. His approach was to look at the biochemical signatures you would find, say, in breast cancer, and then build diagnostic models based upon the specific biochemistry of those changes. And that was by looking at the different regions within tissues and using that biochemical information. We did something similar by building it from pure biochemicals that we expected to be present within the tissues that we were looking at. Now, the problem with both those approaches, if you build these kind of models, you must include as much information as possible of those biomolecules within the environment that they're actually living in, effectively, to make that diagnostic model super accurate. Now, so you have to make assumptions, and there's always some difficulties there. But it did really inform us about the changes that you see around things like lipid. We've done it in the prostate, for example, as well, around the importance of lipids for driving the carcinogenesis process. And there's been a lot of work on lipidomics since then that has sort of backed up some of these interesting things that we weren't even aware had any relevance to cancer at the time we were doing it, sort of 15 years ago. So there's a lot of interesting work you can do by exploring exactly that. And I think more should be done. Absolutely. I think it's a fascinating area.
Excellent. Thank you. We have another attendee who wants to thank you both for your talks and would like to know from Nick what the main elements that they should consider for their lab to set up simple Raman spectroscopy.
Oh, okay. It always depends what you're trying to measure. So Raman spectroscopy can be done quite cheaply if you're trying to measure something with a high concentration of a solution, and the purer solution, the easier. And obviously, as you get more and more complex biological things, signals tend to get diluted, things get weaker, and you need then more and more expensive kit. And so, whatever happens, you need a monochromatic light source, you need a good spectrometer, and you need a good detector. Now, we can work out lots of different ways of getting the light to and from the sample. But the real issue is how many photons you can collect, because Raman scattering is a weak effect. And I think if you know you're looking at low concentrations, then you need quite a reasonable amount of light. So a reasonable light source and a very good detector. Like I mentioned, the depletion detectors, for example, in the near infrared have been very good for us. But it kind of depends on your applications. But I'm happy if you want to get in touch and try and track me down, I'm happy to give you some direct advice specifically on what you're doing, if that would be easier. No problem with that at all.
Perfect. Thank you, Nick. So to finish off today's Q&A, someone is asking whether you could summarize the key benefits of Rahman for biological applications over other optical techniques such as fluorescence.
Thank you. Okay, so the real benefit of Rahman is the fact that the peaks that we measure are incredibly sharp relative to most other approaches, and that most other optical approaches, and that does allow the measure of the whole range of changes in the biochemistry around, whether it's the amino acids, the nucleic acids, the lipids, the proteins, the conformations, etc. I mean, the whole gamut is there, and that fingerprint, the combination of those changes, can allow you to measure both the tissue type, but also the pathology of the cells, and potentially the prognostic information that drives that carcinogenesis process or disease process that may change the structure later on, which is something maybe the clinicians would be seeing looking at biopsy samples. So the real thing is the fact you can provide a real-time measure of the molecular composition of something, which is incredibly powerful. But as I mentioned before, the weakness is its relative signal is very low, so imaging is difficult. So if you're looking at large areas, that's tricky. So you ideally want something that will help you direct your Raman measurements to a particular area, and then you've got a lot of power in the information you can obtain.
Excellent, thank you. So unfortunately, that's all we have time for today. So a huge thank you to both Francesca and to Nick for today's informative presentations, and thank you to everyone who's joined us online. I can see there are still lots of questions coming in, which is great, and we will follow up with you after the webinar, but also please feel free to email me at editor@selectscience.net with any further questions following this event. If you would like to share your attendance or provide feedback using social media, please feel free to mention us using @SelectScience or #SelectScienceWebinars. Remember, you can also download your certificate of attendance in the related resources tab. And if you would like to listen again to today's webinar or invite a friend to listen, it will be available to watch on demand in a few days' time. So goodbye. Thank you to Nick and Francesca, and thank you once again for everyone joining us.
Thank you. Bye-bye.
Bye-bye. Thanks all.